This is Dr. Vliet. Welcome to America's Fabric. Follow me on Twitter at Health and Sense or my medical website, www.herplace.com. We have with us again today my special guest, Dr. Steven Hatfill, an American physician, scientist, virologist, and bioweapons expert with a background in military special operations and training in emergency and tropical medicine and pathology. After serving as an overwinter physician in Antarctica, he obtained separate master's degrees in microbial genetics, radiation biochemistry, and hematological pathology, and has published numerous scientific research papers. He was also awarded the U.S. Army Parachute Wings in 1975 and the Rhodesian Army National Defense Medal in 1979. His postdoctoral fellowships include Oxford University, the National Institutes of Health, and the National Research Council, where he studied the Ebola virus at the U.S. Army Institute for Infectious Diseases at Fort Detrick, Maryland. In 2000, he was certified by the UN as an Iraq weapons inspector. He is currently an adjunct assistant professor at a leading university in the United States and operates also a remote jungle training facility to test new equipment and to train other scientists to conduct research in high biodiversity areas. For the last 7 years, he has trained U.S. Army, Navy, and Marine Special Operations units in tactical combat casualty care. He is speaking to us today on America's Fabric as a board member of Doctors for Disaster Preparedness. Welcome again, Dr. Hatfill. It's a pleasure to have you back with us.
Thank you very much.
Today, we wanted to talk further about some of the treatment issues with the emerging viral illnesses like Ebola and the other hemorrhagic fever viruses. Our focus today is specifically more on Ebola because there's exciting news to report that there actually has been a drug credited with potentially saving the lives of the 2 American missionaries infected with the deadly Ebola virus. And that drug has not yet been tested in humans, but was on an emergency basis used for these 2 patients. It has an interesting story. It actually wasn't manufactured, but grown. The drug is derived from genetically modified tobacco plants. And I find this really fascinating. The tobacco plants are infected with the protein, and then the plants reproduce it like a photocopier. The desired proteins are extracted from the plants and purified into a serum. The drug actually has been developed by Kentucky BioProcessing and a small pharmaceutical entrepreneurial company in San Diego, Mapp Biopharmaceuticals. And the fascinating combination here is that the new medication called ZMapp has actually been showing very positive results in some of the primate testing at the U.S. Army Medical Research Institute of Infectious Diseases. In a study published last year, scientists reported that 43% of infected non-human primates recovered after receiving the treatment intravenously, even though it was up to 104 to 120 hours after infection and after symptoms had developed. So there's a great deal of promise here, and the medication ZMapp is actually a mixture of 3 monoclonal antibodies. And here to explain in common sense layman's language for all of us to understand better how such new medications work is Dr. Steven Hatfield. Welcome.
Hi, thank you very much. The monoclonal antibodies is a very exciting story dating back some 30 years. In fact, I believe there was a Nobel Prize awarded for the discoverers. In the old days, we used a type of human tumor cell, and these are antibodies. They're human antibodies, but they're very, very, very specific for certain proteins. And we go back to the lock and key theory. If an antibody, being the lock, sees a protein that it fits around, which is the key, and this can be a protein on a human cell, it can be a protein on the outside of a virus, whatever, it will bind, like putting a key into a lock, and attach itself. There's a lot of ways to make these monoclonal antibodies over the years. People have even tried using chicken eggs. The old ways with the nude mice, laboriously removing the peritoneal ascites fluid, have given ways to other methods such as plant vectors and the tobacco plants that you described. These antibodies have made very promising inroads, and there's a number of them licensed in the field of anti-cancer treatment. The ability to bind an antibody to a specific protein on a cancer cell and then have the immune system recognize that cancer cell as being unwanted and clearing it from the body. These are tremendously expensive drugs. I think one of the new ones for breast cancer runs the patients about $9,000 a month. But they are holding great promise for a variety of areas in medicine. When we're talking about Ebola treatments, it's probably a good idea to go back and kind of look at the development of some of this stuff. Without specific viral treatments, we're basically down to supportive therapy for these infected patients.
And that's what we've been limited to up until now, correct?
Right. This is efforts to maintain their effective blood volume and blood pressure, the balance of the different salts in the bloodstream, and their oxygenation of their tissues. About a decade ago, everyone went out and tried this chemical called Interferon. It's made by the body. It's very human specific, but it really didn't show any effect on the Ebola virus. Then they progressed to trying to give what we call, the technical name is nucleoside analogs. But these are false chemicals we can put into a patient that we hope that the virus will take these false chemicals up as it tries to replicate its own genetic material and turn into a non-infectious form. And some studies were done with this, but they found it was only sort of effective in the animal models if you could get it into the patient within 30 minutes. Then we looked at what we call the pathogenesis inhibitors, looking at specific sites of how the virus causes disease and trying to interfere with that. And about a decade ago, scientists tried using this nematode anticoagulant protein. This is the protein inside leeches that stops your blood from clotting. And you think it might be paradoxical that you're giving an anticoagulant to a patient that has a hemorrhagic disease. But the thought behind it was to interrupt the vicious cycle that develops during infection. And again, these non-human primate models showed a 10% survival rate. And the fatal monkeys showed a length of the course of their disease by 2 days over the controls. So these were looked at very carefully, and the work progressed a little bit more. And we started looking at the patients that survived. And what we found is that they had very high levels of chemicals called the interleukins, specifically interleukin 6 and interleukin 8 in the fatalities. And that these fatalities showed a deficient humoral and antibody response.
I mean, the people who died had the higher levels or the lower levels, if you could clarify?
Well, the people that died had the higher levels of inflammatory chemicals and lower levels of antibody.
Okay. Just wanted to be sure our listeners understood.
Sorry, I may have dribbled that out. Anyway, so this spurred things on to what were called CPG immunomodulators. And Dennis Kleinman at the FDA was a very leading proponent of this. And these synthetic oligonucleotides, what they are is tiny little strands of synthetically made DNA in the laboratory. And when these are injected into the into an experimental animal, the whole immune system gets turned on. And we tested this stuff in guinea pigs and in mice adapted Ebola that Mike Bray at the National Institutes of Health had developed. And we found if you injected it within sort of like the first 12 hours after giving these animals a lethal challenge of Ebola virus, they didn't even get sick.
That's amazing.
Their fur didn't even ruffle. And all the control animals died, of course.
Well, and one of the things you made, a point you made in an earlier show, is that with Ebola, it only takes 10 viral particles, which is minuscule, to initiate an infection. So the fact that these monkeys showed two entirely different responses is quite amazing.
Oh, yeah. Then we went on to the vaccines. And USAMR was able to get quite a good recombinant vesicular stomatitis virus. This is a virus that likes to attack a lot of different types of human cells. But they took one of these neutered viruses and genetically spliced in the outside protein on it was on the Marburg virus. And they found that it really gave very, very good exposure if you gave the vaccine within 20 to 30 minutes after exposure. And then they worked on an Ebola, and it was found to protect about 50% of the monkeys against infection. So this was all exciting stuff, and it pointed out the role between the central role by these macrophages in the body.
Well, and the thing that I find really striking is that in a disease that carries a mortality rate or death rate anywhere from 40 to 90%, to have something that gives a 50% improvement in survivability is pretty significant.
You know, in medicine, if we see something that gives a 4% to 10% improvement, we get all excited. Right. So, yes, you're right. So 50% is pretty amazing. This is major. And again, these drugs are exceedingly expensive and laborious and time-consuming to manufacture these monoclonal antibodies. It was just fortuitous. I believe that this had been, the FDA allowed it to be an investigational new drug. And I know they were getting ready for phase 1 trials. But I think it was really an act of mercy that it was allowed to be given to these two doctors. Well,
I agree. And the thing that I think is really incredibly interesting as a scientist is that because tobacco plants readily pick up genes inserted into them, this facility in Owensboro, Kentucky, has actually been using that characteristic of tobacco plants to develop other products. Kentucky BioProcessing has done work on some of the biggest health threats on the planet. One of the things that they've done is in being involved using the tobacco plant model, working on the H1N1 or swine flu vaccine, the human papillomavirus vaccine, for example, the brand name is Gardasil. They're working on an anti-rabies antibody, norovirus or the cruise ship virus, and HIV.
Exciting work.
It's very exciting work.
Especially, our biggest threat is still pandemic influenza. I think most scientists will agree with that. And we have to be very cautious how this vaccine is made. And the FDA certifies fertilized egg culture. And the problem is, by the time you've made enough vaccine, the pandemic's already underway.
Yes, and you're almost through that particular flu season by the time they get figure out what they're going to use.
Or that. So to have a way, and anything can be industrialized and made into large batch productions. We have very smart people that know how to manufacture things in this country still. This really requires more interest by everyone. And
you mean the tobacco, using the tobacco plant?
Yeah. The technology has been around for years. They use a shuttle vector to carry little pieces of DNA into the tobacco plant DNA. And those genes can be anything you want to clone in there. But very, very exciting technology, and it sounds like these scientists have really got it down to quite a good art. And I'm hoping that this will make eventually a dramatic improvement in the way that we can quickly make flu vaccines to meet an emerging threat.
Well, that's exactly right, because the method of using the egg has always been one of the stumbling blocks to getting it done more expeditiously in time for when it was needed.
Well, we also had some problems with that, and the fact the H5N1, I think it was, flu virus, it was killing the chicken embryos before it could make a high concentration of virus. And we're talking hundreds of thousands of fertilized chicken eggs.
Right.
So this was always a background worry for this type of thing. So I have great hopes for this technology. And the fact, I mean, once you start the characteristic maculopapular rash in Ebola, that means the virus has gone systemic, it's in your skin, it's spread to your other organs. If you're not able to mount an effective immune response, and only a number of patients are, then that's pretty much the end of you. You go into organ failure, and that's it. So to actually have such an effect at the time of the definitive symptoms and the fact that the virus was spreading through the bloodstream, this is really very, very exciting.
Well, and would you expect the rapid response that we're told that Dr. Brantley had? It seemed like it was actually very, very quick after he was given the experimental drug that he really started to improve.
I was a little surprised. This is sort of indicating to me that small blood vessels in the body are a critical factor in the pathogenesis of the disease. Small blood vessel diseases, we have a number of them. In fact, diabetes is actually a small blood vessel disease.
Yes, you're right.
It's just the high blood sugar, chronically uncontrolled, causes it. This is very, very exciting information. A number of researchers have shown that these endothelial cells, these are the cells that line your small blood vessels, play a central role in the causing of death in these patients and the development of the disease. So,
well, I am extraordinarily encouraged by these developments. And actually, what's also, I think, from a humanitarian standpoint, what's encouraging is if our technology, using something relatively inexpensive to grow as tobacco, means that we may have a way of developing these medications in greater quantities at lower cost, and can once again continue to be one of the things that the United States has been quite strong in, is innovation in medications. And we lead the world in launch of many new medications, particularly in the field of cancer treatment. So I think if this is as compelling as it appears for the Ebola response and improvement relatively quickly, to be able to develop it and help these countries in Africa where the hemorrhagic fevers are so widespread, would be an extraordinary accomplishment.
Yeah, and it brings up an interesting point. Our nation, really, for the last 100 years, has been one of benevolence and trying to help others.
Yes.
And we have dumped billions and billions of dollars into Africa, trying to improve the health and the welfare of the people there. And these leaders, for the most part, have just stolen the money. Nothing's changed. These countries, a lot of them are still a mess, and they hate us. So I can't figure this out.
It's really tragic, and you're speaking as someone who spent a lot of years of your life on the ground in Africa.
Over 15.
That's really extraordinary, and I commend you for your work in this area to better their lives. That's one of the things that has bothered me so much, is to see the amount of money and time and effort and the caring and the concern and compassion of Americans who do a great deal to help improve the quality of life and the health in Africa, and to see that the corrupt political environment takes the money and effort away from the public.
Well, yeah, Nothing has changed over the last 50 years. In fact, a lot of countries have gone backwards. So Americans are caring, compassionate people. And we demonstrate this again and again and again and again.
It ties into...
It's just beyond belief.
Well, I was very pleased to see Dinesh D'Souza's film and book, America, which does talk about the generosity and the efforts and the compassion and the giving and the charity of Americans and our country in general. When you look at the tsunami, for example, that hit Phuket, Thailand, and our military vessels that could get there quickly and give humanitarian aid, when you look at how our technology has been used for good in so many different ways. And I really feel it's important to help people understand the good that America has done. We get so much criticism from so many sources that it's time for us to say, no, that's not the whole story. We're a country of good people who care about doing good for other countries. And
absolutely. I was in Sri Lanka right after that tsunami, and it had devastated the coastline from like Colombo down through Hikkaduwa. I was working in the area where the big train crash had happened and all those people died. And do you know, by the time we got there, the USAID agency and the Marines and everything had opened up that road all the way they just suddenly arrived overnight with bulldozers, cleared all the debris out of the way, and opened it up so the British and other aid agencies could get in there.
And you never hear that in our media.
You never hear it. We are a compassionate people, and we've demonstrated it again and again and again.
Well, I certainly appreciate all that you have done over a very distinguished career to not only put yourself in areas of risk and hardship and sacrifice to be there to do this work, but also all of the effort that you've done to teach and train other scientists, military people, so that they have the skills that they need in the event of disaster. And today, the whole focus of Doctors for Disaster Preparedness is also an effort to educate others, to plan, to be proactive. I think before we close today, I'd like for you to tell our listeners a little bit about the organization and what you're doing and how you're working as a group of physicians to enable better preparedness for all kinds of disasters.
I became interested in the organization because it has some very well-known scientists that belong to it. And these scientists are dedicated to looking at the scientific data and making their own conclusions. And sometimes these conclusions are in direct opposition to popular thought or government programs. They're a very suspicious group, and they stick to the published data. But I just like that approach. You know, we move forward as a species through the scientific method.
Yes.
That is, we have a hypothesis or an idea that maybe this is how things work. We do an experiment, and if the experiment works, we should be able to make a prediction. We do more experiments, and sometimes our ideas are crazy and not right at all, and sometimes we're on the right track. But it's this scientific progress that has allowed us to move forward in all branches of science and technology, and it contributes. Your findings are reviewed by other scientists in peer-reviewed journal, so that what's put out there should contain no ambiguity, no misconceptions, a peer-reviewed body of knowledge. I've seen too many cases where even scientists don't really look at the data and veer off into things that aren't supported.
Well, actually, Dr. Hatfill, I couldn't agree with you more. That has been the story of my career in both men's health and women's health, because the data absolutely contradicts what the guidelines have been so limited and flawed in the government guidelines. And I've done lots of articles and talks and radio shows on that whole issue. So I think you're right. I think that's a very critical point. If we look at the data, we look at how to adapt the data clinically for the best use as we apply it to individual patients. To me, that's the art and science of medicine.
It's evidence-based decisions.
Yes, but not in the way it's currently used, ignoring some of the evidence.
I have to agree.
So
stick to the peer-reviewed scientific data.
Absolutely. And so that's the goal of Doctors for Disaster Preparedness. That's the goal of my work in men's health and women's health. And I want to thank Dr. Hatfill for joining us once again today. He's been very generous with his time to help educate us on these critical issues. Thank you so much. And we'll look forward to having you back on another one of my radio shows in the future. For now, this is Dr. Vliet signing off for today. Thank you for listening to America's Fabric, and follow me on Twitter at Health and Sense, and check my medical website, www.herplace.com. Have a great week, and we'll be back next time.